Welcome to the deep dive. Today, we're digging into some really interesting work coming out of the University of Tokyo.
Yeah, it's looking at how obesity changes the way the liver responds when you're starving.
Exactly. And it's based on a study by Kigo Morita and Shinya Kuroda, published in Science Signaling. That's a pretty serious journal for this kind of molecular biology stuff.
It really is. And this is, fundamental. Our bodies have to adapt when food isn't around and the liver is it's the absolute core of that process.
Right.
And with obesity being such a huge health issue, figuring out how it messes with this basic survival system is incredibly relevant for basically everyone.
Totally agree. And what I found particularly cool about this research is the angle they took. It's not just listing molecules,
Yeah.
They really focused hard on the timing. Like, when do these things happen inside the liver cells?
Which is a really fresh perspective, actually.
Yeah. And it leads to some pretty surprising conclusions, as we'll see. So our goal today is to unpack this study, understand how obesity seems to temporarily knock the liver's starvation response out of sync at a molecular level.
But without causing permanent structural damage, right? That's a key point.
Exactly. That's what makes it so intriguing.
Okay, so maybe let's start with the liver's basic job during starvation. It's not just sitting there waiting, is it?
Oh, absolutely not. The liver's a metabolic powerhouse, always working to keep things stable, that homeostasis idea.
Right.
But when food gets scarce, its role just ramps up massively. It's like the conductor of an orchestra, deciding which energy stores to use.
And critically, when to kick off different pathways.
Precisely. It's all about synchronization, what happens, and just as importantly, when it happens.
Makes sense. But trying to study biology playing out over time, that sounds incredibly tricky. How do scientists even start to untangle all that data?
Yeah, you've hit on a major challenge there. Just imagine tracking all these molecules changing second by second, minute by minute.
It's a mountain of data.
Right. And pulling out the meaningful patterns, the actual signals from all that noise is really difficult. This research actually represents a pretty neat step forward in figuring out how to do that kind of time-based analysis.
Okay, so how do they do it? What was the setup?
So the researchers had two groups of mice, healthy ones and obese ones, and the key was they didn't just look at one point in time.
No snapshot.
Exactly. They analyzed liver samples during a 24-hour starvation period, but at multiple points.
Like checkpoints.
Yeah, at 0 hours, then 2, 4, 6, 8, 12, 16, and finally 24 hours. It basically let them build a molecular activity timeline.
Okay, so comparing the healthy and obese timelines, what popped out first? What were the obvious differences?
Well, one of the early findings involved what they called hub molecules. You can think of these as like major crossroads in the liver's metabolic network.
Controlling lots of different reactions.
Exactly. And in the healthy mice, during starvation, key hubs like ATP and AMP, the basic energy currency molecules were present and active.
Right, makes sense. Energy management central.
But in the obese mice, these specific hub molecules were notably absent or much less prominent at certain points during starvation.
Missing those key energy regulators sounds pretty serious. Like a fundamental breakdown, maybe?
Yeah, that's what you might initially think, right? That something in the liver's basic molecular wiring is broken or damaged in the obese mice.
Seems logical.
And this is where it gets really interesting. When they dug deeper, they found the underlying structure, the actual network of connections between molecules, was still pretty much intact in both groups.
Wait, so the machinery itself, all the parts and connections, they were still there?
Essentially, yes. The framework was okay.
But the response was still clearly different in the obese livers. How?
It came down to timing. The big disruption wasn't what molecules were there overall, but when they became active and how their activity was coordinated over that starvation period.
Okay, so it's not a structural problem, but a timing, a synchronization problem.
Exactly. It's like the orchestration was off.
Right. So in the healthy livers, you mentioned those hub molecules, ATP and AMP.
Yeah.
They responded quickly.
Very quickly, yeah. As soon as starvation began, their levels changed rapidly, showing this really tight control, this precise temporal order. The network knew exactly when to bring them into play.
Like clockwork.
Pretty much.
And in the obese livers, that clockwork was just gone.
Well, yeah, that coordinated timing seemed to disappear. The hub molecules weren't showing up and reacting in that same fast, organized way. The signals were getting sort of muddled or delayed. You had that great orchestra analogy earlier. Can you bring that back? I think it helps clarify this timing issue.
Sure. Think of the healthy liver responding to starvation like a really well-rehearsed orchestra.
Okay.
All the musicians, the molecules, know exactly when to play their part. They come in on cue, perfectly synchronized, creating this, harmonious metabolic response.
Right.
In the obese liver, it's more like, well, the musicians still have their instruments. They might even know the notes, but the conductor's signals are messed up, or they're just not listening properly. They're not playing together in time.
So the right notes, maybe, but at the wrong time, creating noise instead of music.
Exactly. The overall performance, the metabolic adaptation, is disrupted because the timing is all off.
That really does paint a clear picture. So this study isn't just important for understanding obesity's effects, but also for the way they figured this out, the methodology.
Absolutely. That's a huge part of it. The real methodological innovation here is combining that structural analysis, looking at the map of molecular connections.
What's there?
Right. Combining that with the detailed temporal analysis, seeing how activity changes and coordinates over time.
But when it happens?
Precisely. They managed to look past the static picture and capture the sort of dynamic dance of the molecules.
And this approach, combining structure and time, they think it could be used elsewhere in biology.
Oh, definitely. They specifically mention applying it to other OMS data.
Like the genome, microbiome.
Exactly. Genome, microbiome, proteome, any complex biological system where things change over time. If you want to understand not just the parts list, but how the system behaves dynamically, this kind of combined approach could be really powerful. It opens up a lot of doors, potentially.
Super interesting. Do we know what the researchers themselves are planning next? Where do they go from here?
Well, they want to see if these ideas, this interplay between structure and timing, apply in other situations.
Like not just starvation.
Right. Like
Yeah.
what happens during normal food intake or in the progression of different diseases. Can they generalize these findings about temporal dynamics being key? That seems to be the next step.
Okay. So bringing it all together, what's the big takeaway for how we should think about metabolism?
I think the really powerful point is how much this highlights the intricate and time-dependent nature of how our bodies manage energy.
It's not just about having the right parts.
Exactly. Knowing which molecules are involved is crucial, obviously, but it's only part of the story. We also need to understand the precise timing, the sequence, the coordination.
And for obesity specifically.
For obesity, it suggests, at least in the context they studied, that the initial impact on this starvation response might be more about throwing off the timing, the function, rather than causing immediate, irreversible structural damage to the liver's molecular network.
A functional disruption, not necessarily a structural breakdown.
Yeah, temporal vulnerability.
Yeah.
And shifting our focus to include that dynamic, time-based element feels like a really important advance.
Okay, so if we had to really nail down the key message here, it's that obesity seems to temporarily disrupt the timing and coordination of the liver's response to starvation. The underlying molecular machinery, the parts, are mostly still there, but they're not working together in that perfectly timed sequence needed for a healthy adaptation.
You got it. That's the core finding. And recognizing this temporal dimension, well, it has really broad implications, doesn't it, for how we study health, disease, maybe even aging.
It really makes you think, if timing is this critical for the liver dealing with starvation, where else might this concept of temporal coordination be absolutely vital?
Right.
Could subtle disruptions in biological timing be a hidden factor in, say, how effectively drugs work or how we recover from stress or maybe even how our brains learn?
Yeah, what other complex systems in our bodies rely on this delicate internal clockwork, and what happens when that clockwork gets knocked out of sync? It's a fascinating question to ponder.